In this video, we shall examine the question, is there or has there ever been a Mercury biochemical cycle on the planet Mars? Why is this important? Why should you care? Well, if you're interested in finding evidence of extraterrestrial life, I would encourage you to grab a pen and a piece of paper and stick around for the whole video. Before we go to Mars and contemplate the possible nature of the cycle of the element Mercury, let us first discuss what this natural cycle looks like on this planet Earth. This is a highly simplified Mercury biochemical cycle. Mercury is naturally released into the atmosphere by volcanic emissions. Once in the atmosphere, it is carried to land surfaces and aquatic ecosystems, where it enters the water column and migrates to sediments. Sediments tend to be anoxic, and in this environment, inorganic mercury is converted to an organic form called methylmercury by specific anaerobic microorganisms. This process is called methylation. Some of the methylmercury binds to particulates in sediments and is buried, while some of the methylmercury diffuses into the water column where it enters biota, making its way into fish. The methylation process not only occurs in aquatic sediment, but also happens in soils. So to summarize, the aspects of this cycle that are relevant to our discussion of Mars are volcanic emissions, atmospheric transport, moisture either in soils or aquatic sediments, and the methylation process, which is the conversion of inorganic mercury to methylmercury by anaerobic microorganisms. This leads me to the point of this video. I want scientists all over the world to take Mercury biochemical cycling very seriously, as it has the potential to answer one of the most fundamental questions, which is there or has there ever been life somewhere other than our planet? I would argue that in the case of Mars, it makes a lot of sense to look for the presence of methylmercury as an indicator of past life. Unlike methane, methylmercury is not volatile and can bind to particulates and sediments, leaving it immobilized. After an environment that once favored but is now hostile to life, long after cells have died and their various components degraded, long after methane and other gases indicative of life have diffused away, methylmercury made by microbes and absorbed onto particulates can serve as tangible evidence of past life. Another feature of methylmercury that makes it desirable as a biosignature is its ubiquity. On Earth, methylmercury is everywhere. Coastal sediments and tidal marshes are methylation hotspots. But methylation is not limited to these locations. When I say everywhere, I am not joking. Go outside your house, take a teaspoon size of soil and analyze it. There is methylmercury in that sample. Go to remote areas on our planet, areas where there is no history of industrial activity, like the tundra of Alaska and the continent of Antarctica, and take soil or sediment samples. You will find methylmercury. So, why is methylmercury in measurable quantities everywhere on this planet? Two reasons. The first is the high and widespread availability of the substrate inorganic mercury due to volcanic and industrial contributions to the atmosphere. The atmosphere is a very efficient global transport and distribution system. The second reason is that microorganisms that make methylmercury from inorganic mercury are ubiquitous, extremely tough and resilient. Some can survive in a variety of environments as long as there is water. While they can be abiotic methylation, that is methylation that is not biological, the methylation process is primarily and overwhelmingly driven by anaerobes in soils and sediments. Now, let's go over to Mars to see if these factors that favor widespread methylation on Earth exist or have existed on Mars. When we look at Mars today, it barely resembles our planet Earth. Even though we do not see active volcanism today, there is evidence of extensive volcanism in the past. Such evidence includes channels made by lava flow and notable volcanic structures such as Olympus Mons. Mars has an atmosphere, Hence, I expect that past volcanism released a lot of mercury into the atmosphere, which was then distributed planet-wide. While we do not see bodies of water on the surface of Mars, it is widely believed there was liquid water flowing on Mars in the distant past, based on the presence of stream beds, river channels, and rocks on the surface that only form in water. Furthermore, 3 underground lakes have been discovered in Mars in recent times. So, there was volcanism, there is an atmosphere, there is evidence of past water on the surface and current water underground. All that we're not sure of is the presence of microorganisms that methylate mercury. So, let's talk about that. Some scientists are actively investigating whether observed methane measurements and seasonal fluctuations in Mars are of biological origin. The microorganisms that produce methane on Earth are called methanogens, and some methanogens are known to produce methylmercury. In fact, all groups of organisms that produce methylmercury on Earth, sulfate-reducing bacteria, iron-reducing bacteria, methanogens, some firmicutes, are also thought to be capable of surviving underground in Mars if there is liquid water. Furthermore, we know that sediment and rocks can move from world to world due to asteroid impacts. We have found several rocks that came from Mars on Earth. Is it that much of a stretch to suggest that rocks and sediments could have traveled from Earth and transferred life to Mars in the distant past? Nevertheless, until we get confirmation, this is only an educated guess. What got me thinking about this is the recent news that Perseverance rover will collect surface samples from Mars for a future vehicle to return to Earth. Knowing this, the question is, what kind of tests will scientists perform with these samples? Is Perseverance able to collect subsurface samples? If yes, have the research team considered mercury speciation analysis in subsurface samples? To summarize, given past resemblance of Mars to Earth in terms of volcanism, a distribution network in the form of an atmosphere, and water previously on the surface but now underground, it is my argument that scientists should at the very least consider analyzing surface but preferably subsurface soils and sediments from Mars for mercury speciation, as it has the potential to help in the search for past extraterrestrial life. So, what do you think? Since NASA is bringing back samples from Mars, what biosignatures in either surface or subsurface sediments would you look for? Please let me know in the comment section below. Upcoming videos include the case for regular and routine sample return from Mars and other interesting locations in the solar system in the search for current or past extraterrestrial microbial life. UD out.